Now let's define a new function to add proteins to RNA UMAP. To RNA UMAP. I'd like to have high variable genes, RNA, and our data frame for proteins. So first let's get a log version of our proteins. Log 1. Okay, so this is gonna make things easier to visualize, and I'm gonna find common cells between RNA and proteins. So we're gonna go to observation names, intersect. I want to make sure if the rows are aligned correctly. Okay. I'm gonna keep only the shared cells inside our RNA. We're gonna keep only the shared cells in protein table. So now I have to go through a loop over all the proteins and add one protein as a new column in RNA observations. So for protein name in log.names, columns, I'm going to make a new column called protein name, and then I'm going to assign the name of that protein. Values. So now ScanPy can color UMAP by that protein. And so we're going to return and protein looks. All right, but before we call this, I'm guessing I would come here and return this RNA high variable, because later we have to pass it to this function. So I'm gonna go back and assign it to RNA high variable gene, and then we're gonna import this guy. So I'm going to pass the protein real and then later call that to data frame function right here, if it is here. Is it already? Yes, okay, it's already a data frame. So I don't need to do that. Maybe it's best if I call this here. All right. Now let's try to plot this. So I'm gonna grab the first, let's say, 6 proteins alphabetically. And then we're gonna plot this. And we're gonna add selected proteins. So the color map would be so that will give us a purple to yellow. It's default, and it's a good choice. And I'm going to return the selected proteins. But also I would like to this should be data frame, and I'm going to call this function right here. All right, looks good to me. And so I am going to save this. Now let's run. All right, so let's take a look at. All right, so we're gonna start by CD80, which is the top row, second from left. And as you see, these dark purple points are representing almost low or zero expressions. And the more yellow they become, it means they have higher expression. So almost everything here at CD80 is dark purple, near zero everywhere except a few scattered, bright cells like here, or in this isolated cluster 6 that we had. Yeah. And so if we remember the sparsity amount that we had. Yeah, so CD80 had 20% sparsity, meaning 20% of cells have 0 CD80 in them, which means 80% of cells do have some CD80. But the UMAP is showing us almost everything near 0. But that still makes sense because the sparsity counts show the exact zeros. So a cell can have a non-zero count but still appear dark on the UMAP because the value is so small. It is essentially a background noise, not real CD80 expression. Sparsity counting exact zeros underestimates how sparse a protein really is in practice. Many cells have counts of 1, 2, 3, which are technically non-zero, but biologically meaningless. Their logged version, like log of 2 is 1.1, right? Still looks dark on the UMAP. This is why looking at both sparsity plots and UMAP together gives you a more complete picture. So let's look at the third panel for TIGIT. So this scattered yellow-green dots spread across this left blob and parts of the middle one here. And so no single cluster lights up cleanly. I did a research on this specific protein, and it looks like it's an exhaustion activation checkpoint marker on T cells. So it should be expressed on a subset of T cell across multiple clusters rather than defining one specific cluster, which makes sense here, right? Because this cluster, which is 4, has both yellowish and some dark purple here. So it's not taking a whole cluster, it gets a subset of it. And so this will be a harder protein to predict because the signal is kind of noisy and diffuse. And let's move on to the next panel. So we see cluster 4, right? This is 4 and this is 8. So 4 and 8 are light up. It means there are high values in there, and then we have this middle blob that is kind of like in the middle one, and then the rest of them are kind of darker. So the model has a clear gradient to learn about this CD16, so that's good. And moving on to CD137, looks like it's a very near zero variance target, because as you see in every part of the plot we have like dark purple to yellow, or like it's mostly teal. It's like you see combination of everything at the same time everywhere, so no cluster is standing out. The model has almost nothing to learn, and so predicting the mean would be almost as good as any complex model. This is what low variance look like in the variance plots we saw earlier. Now we can see visually confirmed, right? Yeah, it's a very low variance. So let's move to CD274. Yeah, again, very low variance here. And CD19, this is, so we have one single cluster here that is all white, and then the rest is almost same. This is really good because perfectly separate, these targets for a cluster. If the model learns which cluster a cell belongs to from RNA, it can predict CD19 almost perfectly. So high signal to noise ratio, this is our best signal. So by looking at all this, I would say as we go forward, I would train one model based on all the proteins together. I won't train a separate model for each of these proteins. I'm going to train one model for all the proteins. But I would like to evaluate pair protein performance separately. So even though you train on one single model, you should evaluate each protein separately. And of course, we don't want to just get the average performance and judge the model based on that, because it could work on some proteins very well, but not others. So that's going to be the strategy forward. So that's it for this video. In the next one, we're going to build another UMAP based on gene-protein pairs that we know of, the most common ones. So thanks for watching. I hope you enjoyed coding with me.